Okay, thank you very much for the opportunity to talk about our little company here that you may find can relate to many of the different projects we heard about since this morning. e-Zyvec is a small service company, young one as well. It's a university spin-off, 3 years old, and 8 people only so far. We're based in France, in Lille, in a little garden between London, Paris, Amsterdam, and Brussels, which is a very nice playground for biotechnologies and biologies in general. So it's a very good place to be. What we do is actually one thing mostly is assembling DNA fragments to create expression vectors. So good vectors, most of us online, we know them for a while because they existed when we started our careers in science, and basically it's been 70 years that we're using molecular tools like that, expression vectors, to perform genetic engineering for multiple purposes. One is actually increased knowledge, and that's all the actions related to fundamental research. But more and more they use as well in bioproduction for drugs, but also for industrial biocompounds. And DNA vectors are even used as drugs directly, like in gene therapy, for example, with viral vectors, for example. So there's many different applications, and we're all very familiar with that, and we all think that it's a very trivial question. But actually, the thing is, in 70 years, the people who actually develop these fields, well, they went to retirement, most of them, and the knowledge and the know-hows are actually leaving the laboratories progressively, while the numbers of users and the numbers of needs are increasing. And nowadays, it's actually taking quite some time to just produce the tool we use, while the value is actually using the tool. It's a bit like when you're talking about your garden, and you ask your gardener to perform a nice project, and he starts building his tool with piece of metal and wood. Of course, at the end of the day, the garden will not have progressed very, very quickly, because his actually job, his work is to use the tools. He will know what is the good tool to use, but not necessarily what is the best way to create that tools. And this is what we do at e-Zyvec. We are the tool makers for the biologists, so they can actually focus on more high value tasks in their projects, whatever the project is. So let's say, for example, today you want to produce a viral protein to make a serological test or vaccines. Or some vaccine. When you start to think about the rationale of your project, you would have some coding sequence of interest to integrate or to insert to a vector, and maybe you have some variants of that, of course, and you would like to tag that so you can peripher it or follow it in a biological system, and you want to, of course, express it. So you need some promoters, and you will have some signal peptide to use, so you can increase your rate of productions. There's so many parameters you can play with, and that's just one example amongst many others. And so many parameters for each category, that if you want to actually do the full study in a very rational way, we just had a very large study just before, it becomes creating a lot of expression vectors in one go. And how can we do that in a very fast way, being accurate, not missing any of the combinations, and still remaining cost effective? And this is what we try to bring to the field. What we do at e-Zyvec is actually not starting with the main of available for your study, but really starting with the whys and hows of the experimenters. So the idea is we start asking, what do you want to see, what do you want to achieve, what is your readout system, and so forth, and let's co-design together the actually genetic tool or molecular tool with all the genetic features that are required for your study and for your particular project, because of course every project is unique. And we start to think together how we can source the different parts physically. Is there existing vectors we can work with or start with? Is there some synthesis to provide and so forth? And then we will be able to, in silico, create the vectors using the proprietary technology of assembling DNA we're using, validate that with the customer, the user of the vector, and build it as we actually produce the maps. So it's like a very big Lego game somehow. We create the parts, we assemble them, and if it's good on paper, then this is what we will do, because as we have created it in silico, we already have set up all the protocols for the assembly. And of course, while we provide that, this work, we check, of course, the vector, the molecules produced are exactly the one we were saying, by sequencing the full molecules with some Illumina platform. So what is nice with this way of working is, of course, you may have different questions and different step projects. And once you've answered your first question, let's say about the promoter you want to use, then you can go back to maybe the peptide signal you want to combine with your promoter and do a new series of vector. And this time it's going to be less questions from us, less design to do, because mostly it means for us to just reintroduce one part in the vector and not changing the rest of it. So it's faster turnover and reduced cost in the second round and follow-ups. So what is different? So you're very familiar, I'm sure, with the cloning by inserting a sequence of interest in a pre-existing vector. What we do, as I said, is constituting DNA bricks that we can assemble in a one-step, one-tube reaction, up to 12 bricks at a time to the letter. That means without any nucleotide scar in the molecules. And what you've got in your rainbow molecule here is actually a lot of controls of all the parts in order and orientations, and also in nature. And if you've got 50 variants of your green bricks, for example, you will just make 50 tubes going to the thermocyclers, each with the same mixtures of bricks, but for one green brick different in every tube. And that is the same assembly protocol we've designed for your series of vector. It goes, as I said, simultaneously in the thermocycler to be assembled in one go. And you already have all your series of rationally designed molecular tools. I want to stress here the fact that we don't do random combinations, we actually do rational combination. We're trying to be exhaustive. So it's a complete freedom of design and very accurate assembly method. So some of you may be familiar with, I'll get with the other part later. So what is different as well in what we do? Do that, we rely on informatic tool, of course, that we developed over the years, which starts with a nice drawing vector platform we just launched last week, where everybody can connect and create vectors in silico by just dragging and dropping sequences from our database or from any sequence you would paste in your own database online on the platform. And this is the first step for us to actually know exactly what is the project about, what are the different molecules to create, and also the software will compute all the sequences once the project is validated to create automatically all the protocols based on the different reaction mixes we use and different protocols we've actually optimized for this. So there's a huge database and growing database with all the parts and vectors possibilities and so forth. And also we last year refurbished and built our own laboratory to actually produce the different combinations of vectors asked for by our customers with state-of-the-art multiplexing equipment for PCR quantification and, as I said as well, NGS sequencing. So, yeah, some of you are probably aware of all the modular assembly method existing on the market. like the Golden Gate, the Mocklow, or in this case here, the Gibson assembly system. And of course, we compared our method to what is existing elsewhere, because it's inevitably some people hear about us and say, well, I can do that myself, and I'm like, let's say I'm using Gibson assembly, I can do it. So we just compare the methods with here 5, 8, or 10 different bricks. They are exactly the same final vectors in both condition, orange and blue. But with our method in orange and the Gibson assembly in blue. And as you can see, the log scale is representing the bacteria with the correct assembly results in the end. And of course, Gibson assembly or other methods are working fine, but ours is more robust, especially when you increase the complexity of the assembly to generate. Now, what is not showing on this graph is the difference between orange and blue is that we do orange and you do blue, meaning your time and your resources are involved in making the vectors from the design to the production, while we provide it as a service. So we do the design and we do the production, and you receive the ready-to-use molecules in your labs. So as I said, you're saving a lot of time for higher value tasks, more related to your core projects. So some key numbers is the foundations. We soon will have created 1,000 of different vectors with only 3 people in the production team, and this is very wide range of different molecules, from viral vectors to bacterial expression or multi-cistronic expression vectors and so forth. It's nearly 100 of projects undertaken for customers, and only 3 times we couldn't deliver some vectors, and actually for the 3 vectors we couldn't deliver, it was all related to the same sequence asked for the 3 constructs, which apparently couldn't agree with bacterial amplification, and we were demonstrating that we could assemble it, but we never succeeded to amplify it in bacteria. So I suppose this is the biological limit for now, but it's still a very, very low rate of failure. And what can we help you with now? Well, virtually everything which is DNA related in your project. Of course, we've been doing a lot of genome editing and CRISPR related vectors with knock-in, knockout projects or imaging projects and regulation of transcription or epigenetic factors projects and so forth. Everything related with protein engineering, and I've got an example later on. We're very good with creating multiple gene expression tools, especially real multi-cistronic with different promoters within the same molecule. And of course, all of that can be used for cell engineering. So just a couple of examples very quickly. Let's say you've got your off of interest. This is a transmembrane protein. For us, it's actually something that we can reassemble, and we can pick what we call the stitches between the bricks everywhere in the sequence. So we can cut it off in different parts and reassemble it as we want. Even if it's a very, very large protein, we've been doing 15 kb proteins this way. We can recreate it and in any kind of backbone. And then you can decide to actually remove some domains just by omitting some of the parts and stitching the full off to the rest of the backbone. You can decide to introduce mutants, single mutants or combinatorial mutants, and the easy way for us is actually to pick a stitch that will create the mutations. So it's very, very easy for us. You can fuse that with everything you want, any position you want, and you can do, of course, combinations of that, fuse some dilatant and fuse with mutants with other fluorescent proteins, and so forth. So once you've got like different chimera, why not putting them into the same molecules? And this is what we call the multi-cistronic vectors. So it took us 2 weeks to assemble a 4 cassette expression vector from 6 different DNA bricks. Some of them only contain 1 genetic features, others are containing multiple ones. And the idea for us here was to actually have 4 cassette driving different fluorescent proteins targeted to different subcellular compartments by fusing that to appropriate addressing peptide or protein domain. So very quickly, it was, so as I said, one reaction, 80% success in assembly. There's the middle one construct is apparently missing one part, but it's not a problem. We've got 4 others amongst the 5 we picked that were okay. And the second one was sequenced and found completely consistent with the predicted sequence. And once transfected into an HeLa cells here, you could see we could light up green at the membrane, blue at the mitochondria, red at the Golgi apparatus, and yellow at the nuclei in one single transfection. So this is a way to show that we can do very tailor-made, personalized construct. So the system is safe. We've got absolute control of the vector content from the full during the full processes. It's open. You're always in charge of the validation. That is a different step. It's new in the way that we assemble things modularly, and we can create any vectors. I used to say that if it doesn't exist, then we can make it. It's easy because you don't need to do anything anymore. We are actually building the vectors ourselves. We try to keep things affordable, of course, and the way we do it is by assembling multiple vectors simultaneously, as I explained. And this is the way we cut our costs and of course your fees. And I forgot to mention that this is a scientist-founded company for several of the scientists. So we're very, very happy when we can actually have a cutting-edge project as well and improve the science through our actions. So welcome to the 21st centuries of molecular cloning with e-Zyvec. Thank you very much. Happy to take any questions.